In this section, we'll be chatting about DNA screening concepts. You'll be using some of these later in the hands-on exercises, and this should help frame how some of this is implemented. I work at Aclid, where we're building security and compliance automation for gene synthesis and governments. We help identify pathogens and toxins in DNA sequences that are either synthetic and natural, and we help industry with customer verifications and compliance checks, really helping automate a lot of the process, making sure they protect their liability and stay compliant. When we think about biological risk, we often use a few buckets. Sequences in the red and yellow buckets typically derive from highly pathogenic agents, while sequences in the green bucket are either common to all of life or are unique to non-pathogens. Sequences in the red bucket are both from highly pathogenic agents and known to directly cause harm, such as encoding a toxin, or are responsible for pathogenicity. For example, invading into a host cell or evading the immune response. Sequences in the yellow bucket similarly derive from highly pathogenic agents, but don't neatly fall into a bucket for pathogenicity or a bucket for housekeeping or metabolic genes. Their function may not be known, or these sequences might be in a gray area where a scientific opinion is required depending on the use case. Sequences in the green bucket, on the other hand, are sequences that are considered relatively safe. These are either housekeeping genes from highly pathogenic agents, so they're conserved, may be found across all of life, or metabolic, or they aren't really found in a pathogen agent and so can be considered to be relatively safe. Some of the most highly pathogenic agents are also found on regulatory lists. Many countries have export controls that govern what types of material can be sold abroad, and countries that participate in Australia Group all agree to control a common set of materials at a bare minimum. Some of these countries include countries in Europe, Asia, and in North America, countries like Canada, the UK, the US, Japan, Korea, and many others. In the US, there's additionally the Federal Select Agent Program that monitors the possession and transfer of certain agents, including some of their DNA sequences, as well as a screening framework by the Department of Health and Human Services that you may have heard about earlier in the course that governs how the synthesis of DNA is done in industry and research. When sequences are flagged due to regulatory or biological risk, it often warrants a review. The review is used to determine legitimacy and what, if any, safety precautions need to be taken. You'll probably want to document the results to reuse or look back later, and these will oftentimes be used for standardization and for helping make sure that you're conducting research safely and responsibly.